Hello, welcome again. This is Physiology, episode 3 about membrane transport. So, membrane transport mechanisms can occur through passive diffusion or active transport. So, the membrane transport here, first we did about passive transport. Passive transport is a transport in which no energy expenditure will occur. Categories that undergo passive transport include simple diffusion, osmosis, filtration, and facilitated diffusion. Example, you can take glucose. So this transport mechanism can occur through lipid bilayer air and through the protein channels. This illustration has shown that a random structure of random movement of molecules and substances in ECF and ICF. Okay, so when you compare the simple diffusion to the facilitated diffusion, as it rate of diffusion versus concentration of substances, so facilitated diffusion will reach Vmax but cannot pass the Vmax, but facilitated simple diffusion will occur continuously, okay, without Vmax. Okay, so the facilitated diffusion could occur through the transport molecules. It will have a binding site in the plasma membrane, so the carrier and the conformational change will occur. Then this conformational change will release the particle or the substance to enter into the cell. Okay, so releasing of binding site will occur finally. So fixed law of diffusion will be applicable for such substances. Okay, this is first law of fixed law of diffusion. So transport pathways through the same membrane, and the basic mechanism of transport will look like this. This is illustrated as simple diffusion. It can simply pass oxygen and carbon dioxide can pass through lipid bilayer. Okay, substance and protein channels or can enter and exit at the same time. So simple diffusion will occur like this, and the facilitated diffusion will occur towards inside or from inside to outside using the carrier proteins. So the carrier proteins also will be applicable for the energy expenditure. Okay, here for active transport, that means so it can enter or it can expel from the interior of the cell to the exterior parts. So these are the mechanisms. So there is also a condition known as bulk transport. It could be phagocytosis or endocytosis. In the phagocytosis, cells use pseudopores to surround the engulfed particles, and pseudopores join, fuse, and surround the ingested particle or the food vacuole. And lysosome digest food vacuoles inside the cell, protect from invading organisms, so remove debris in a way. Okay, endocytosis, or it could be pinocytosis, is one mechanism. Non-specific process, plasma membrane invaginates, fuses, vesicles containing ECF pinches off, and vesicle enters cell into pinocytosis, or also known as cell drinking. Okay. And the bulk transport, there is also receptor mediated endocytosis, which is interaction of molecules in extracellular fluid with specific membrane receptor proteins, and membrane invaginate, fuse, pinch off, and forms vesicle, and vesicle enters into the cell. Okay, in the form of ectocytosis, process by which cellular products are secreted into extracellular environment. It is in which proteins and other molecules to be secreted are packaged in vesicles by Golgi complex inside the cell, and vesicles fuse with plasma membrane and release contents into extracellular environment finally. Okay, this is the bulk transport, extracytosis. You see, vesicles fuse with the membrane, they open to the outside and discharge their contents like this, as diagrammatically depicted. Okay, finally it will be released. Endocytosis, it will be engulfed, it will be endocytosis form, then into inside the cell it will enter like this. Okay, active transport in which a transport that requires energy. The energy used mostly is ATP is used. So because it is active, because it is transported against their concentration gradient, it could be primary or secondary. The secondary is also called called core transport. So the basic route transport for phagocytosis, cell eating, or pinocytosis, cell drinking, and receptor-mediated endocytosis and exocytosis. So active transport. The commonest active transport is sodium-potassium pump, which has a subunit of molecular weight about 100,000 and B subunit about molecular weight of 55,000. So it has its own characteristics. It has 3 receptor sites for the binding sodium ions, and it has 2 receptor sites for potassium ions on the outside. The inside portion has an ATPase activity for the expulsion of sodium against its concentration gradient. So ATP will be used, so adenosine diphosphate and inorganic phosphate will be formed, and potassium will be entered through this mechanism against its concentration gradient because of its energy or energy expenditure. Okay, so all cellular activities or all cells have such mechanisms. So this is outside, this is the inside. Okay, this is how it is transported. Okay, so what's its importance? Because it controls cell volume in its nature, and it favors also for the electrogenic nature from the positivity and negativity of inside and outside the cell. So there is also calcium pump, which is kept low normally. There is also hydrogen pump in the gastric gland, in the late distal tubules and collecting ducts. The characteristics include it can be saturated, it is more or less specific, each has a different affinity, and is inhibited when the energy supply to the cell is disrupted by several factors. Okay. So the co-transport or import of glucose alongside with sodium ions will occur. This is a co-transport or import. Exchanger or antiport couple the movement of two solids in opposite direction, like sodium calcium and sodium hydrogen exchange, or chloride or bicarbonate exchange. This is a counter transport mechanism. So this is a potential mechanism for sodium co-transport of glucose. There are gates also in the plasma membrane. There could be voltage gated and ligand or chemical gated. As you see it in the outside, there is sodium activation, and sodium gate will be open and it will enter into the cell, and potassium in the same manner from the intracellular to the extracellular will live as such in such a way. Okay, the co-transport import movement is in the same direction, so sodium glucose co-transport could occur from outside to inside. This is a import, or the antiport is sodium and solid will exchange in opposite direction. This is secondary active transport. Active transport through cellular sheets, as you see it, sodium and sodium and also water. Okay, sodium and water will diffuse as you see. Active transport through also cellular sheets through the basement membrane. They come from the brush border, so occurs through the intestinal epithelium of the renal tubules in the kidney, epithelium of alexocrine glands, epithelium of the gallbladder, and the membrane of the choroid plexus of the brain, and the other membranes as diagrammatically depicted here. Okay, so transfer of material information across membrane, cross membrane movement of small molecules, diffusion, passive and active facilitated, and active transport. Cross membrane movement of large molecules, these are in the form of endocytosis and exocytosis, and signal transmission across membrane is due to cell surface receptors. Signal transduction include glucagon, which leads to cyclic adenosine monophosphate. Signal internalization coupled with endocytosis, example LDL receptors. Movement to the intracellular receptor, steroid hormones and a hormone for diffusion, intracellular contact and communication. So this is it, energy requirement. Okay, the energy requirement could be passive or active. The active could be in primary active transport or secondary active transport, or in the form of endocytosis and exocytosis. And the passive part is simple diffusion or facilitated diffusion. So the transport mechanism could occur through lipid bilayer, simple diffusion can occur, and through membrane spanning transport protein, including simple diffusion, facilitated diffusion, secondary active transport, and primary active transport, and through the membrane vesicles, endocytosis and exocytosis. Yeah, This is all about this is a brief lecture of episode 3. These are the references I used. Normally the reference we take is textbook of medical physiology by Guyton and Hall. This is the Holy Bible of Physiology. You should read always this textbook. For further information, for further elaboration, you can read Big Picture Physiology by Jonathan and Physiology by Bourne and Levy and Ganong Review of Medical Physiology if you want. And there is also Atlas of Physiology and Human Physiology, the Mechanism of Body Function by Vander and his friends. So thank you for your listening. This is the end of episode 3. I will come back soon by episode 4 about membrane potential. Thank you for your listening. For more physiology lecture, as I told you earlier, subscribe. Thank you.